Hello, everybody. So we're going to cover Chapter 3 today, the cellular level of organization. We skipped Chapter 2 because that's chemistry, and that has a lot more to do with physiology than anatomy, although I wouldn't say there's no chemistry in anatomy. But for what we're working on right now, let's just focus on the anatomical aspect of the body, and we will continue on now with Chapter 3. So cytology is the study of the structure and function of cells. And so one of the reasons I wanted to talk to you briefly about cytology is because it's really important to understand how we look at cells, what organelles are inside of cells, and understand how the basics of how cells are put together, basic cellular anatomy, because it's the cellular anatomy and structure that allows tissues to develop. And we'll get to that in a little bit, but it's very important to understand the structure and general function of cells for this course. Cell structure can be studied using microscopes or microscopy. So microscopy is a discipline where you are looking at really small things. There are basically two different types of microscopes that are used very, very frequently. The most common is the light microscope. The light microscope is what you would be using in lab, and basically what that allows you to do is magnify up to 1,000 times, sometimes a little bit larger, a cell's actual size so that you can see it. Sometimes you can process a cell in such a way that you stain certain components so that you can see them inside of cells and you can see individual structures. Another type of microscope that's used frequently, but mostly students don't do this, you have to have a technician to do this, is an electron microscope. And in this particular case, you are literally using electrons to identify elements of a structure. And there are two types. The first type is called a transmission electron microscope. And what you do is you take a cell, so let's pretend that this round thing is a cell. Let's say this is the nucleus for now. And you slice that cell up, usually in less than or equal to 1 micron sections. And remember, 1 micron is 1/1000th of a millimeter. You put these cells and tissues on a microscope slide, right? So let's pretend that this is a microscope slide. So you would take, let's say, just this piece and lay it out on the microscope slide. So you would have, like, here's the nucleus. Sorry, I went outside. And then you would be bouncing electrons, focusing electrons through this. And basically what you're doing, just like you do with a light microscope, is you would be casting a shadow down below and detecting the shadow. So cross-sections of cells and tissues are looked at using transmission. And we say transmission because the electrons are either transmitting or they're being absorbed by the tissues, just like light. A scanning electron microscope, that's a different deal. That's where you have an entire structure. So let's say, for example, you have like a euglena. That's my version of a euglena. That's a sad euglena. And what you're doing is you're focusing electrons on the euglena, and you're looking at how the electrons basically bounce off the structure, and it gives you the external view of what the cell looks like. So transmission, you're looking at the internal elements. And with scanning, you're looking at external. So we will look at some other pictures pretty soon here, and I'll identify whether it's transmission or scanning electron microscopy when we look at those. We're going to move on now to cell theory. Robert Hooke was the first scientist not to observe cells, but he really wanted to understand what are living things made up of. So he observed cells in actually a piece of cork, right? Cork comes from trees. And he used a very, very early version of a microscope in 1665, okay? So we've had microscopes around since the 1600s, a little earlier than 1665. But basically, what Hooke identified were that cells are fundamental units of all living organisms, all right? So Hooke first identified cells, described them. Other scientists realized that the spaces inside of tissues were the cells, okay? And so cells are the fundamental units of living organisms. They're the structural building blocks of all plants and animals. Cells are produced by the division of pre-existing cells. So cells make cells. Cells just don't go poof and they exist. You must have a pre-existing cell to make another cell. And cells are the smallest units that can perform all vital functions, and I'm going to add here, of life. Okay, cells are the smallest things that can be alive. So here's an example of a light microscope view of the respiratory tract in humans. And then we have transmission electron microscopy and scanning electron microscopy. Each one of these images gives us slightly different information. A light microscope can help you identify much more easily, because it's just light and stains. You can very, very quickly get a preparation, an image of the tissues you're looking at. So what you're looking at here in the respiratory tract are these columnar cells in this region here. Okay, so I'm going to outline a few cells. Underneath those columnar cells are smaller cells. Okay, and they're kind of stacked in here. They're pseudostratified. So here are our columnar cells. But on top, you have this little kind of what looks like a hairbrush, or not even a brush. It just looks like a bunch of, I don't know, hairs. And that's exactly what they are. These are cilia that are on top of the individual cells in the respiratory tract. Now, when you look up closer with an electron microscope, you can see the actual cells, the elongated columnar cells, all right? Here's the cell's nucleus, really well defined. Here are some vesicles. Sometimes you can make out other items inside the cell, like mitochondria and so forth. In this image, I can't necessarily see that. What you're looking at in this particular image is, here's a second cell right here. Now, is it missing its nucleus? No, but what you did was you caught that cell, instead of taking a full section like straight through, you got what we call a grazing section through just part of the cell. However, you still see the cilia here. So you get a really nice internal structure of what the cell, or image of what the cell has inside and on the outside. Right here, who knows what that is? That could be mucus, that could be something that attacked the respiratory tract, could be a dying cell, who knows. Now with the scanning electron microscopy image, you can actually make out all the individual little cilia. Look how beautiful, but you can't see in the cells. So cytologists really spend a lot of time using all three techniques to get a very good visual idea of cellular structure inside of tissues. So let's first talk about cells before we talk about building tissues. Now, there are lots of different types of cells in the body. However, there are two basic types in the body. Germ cells, okay, germ cells are going to be egg and sperm, okay. So here are our germ cells right here. All right, and then somatic cells are all the other cells of the body. Oh, not calls. You know, I need to figure out how to erase. Let's just do this. Cells of the body. Boy, I wish I could write more neatly. I'm so sorry, you guys. So we consider in anatomy, for the most part, somatic cells, because those are the cells that are doing their job in the body. We will talk a bit about germ cells later. Now, somatic cells have all kinds of different crazy shapes. This is a bone cell, an osteoclast. They look pointy. As one specific type of neuron in the brain can have many, many dendrites. This is called a dendritic field. Isn't that beautiful? But one long axon. A fat cell, I actually really like fat cells. I think they're pretty cool. This is all basically an oil droplet. This is lipid, cholesterol, okay? Our fat cells store a huge droplet of oil inside of them. Here's the nucleus. Isn't that funny? Look how small the nucleus is compared to the oil droplet inside of a fat cell. Here is, again, we were looking at cells in the respiratory tract in the last slide. Cells in the intestinal tract look similar with their, over here we have microvilli, not cilia. Blood cells have many different shapes and morphologies. Morphology kind of means shape. And this is an example of a smooth muscle cell. So we have a variety of shapes for cells in the body. So cell structures, the things inside of cells fall within categories. So first of all, you have different cells. We're just going to generalize right now. Within the cell, you have the cell membrane, which is like the cell's skin, okay? And it is made of, believe it or not, fat. Made, ooh, I think I might have found the eraser. Made of lipids. Lipids are a type of fat. The cytoplasm is everything inside of the cell. The cytosol is liquid, and the organelles are actual structures. Now, the organelles themselves are broken down into two groups. The non-membranous organelles, membrane, okay? I am going to spend in a few slides a little bit of time talking about membrane, because membranes and the structure of membranes is tremendously important, and membrane structure allows cells to perform many of the jobs that they perform. Without membrane structure, we wouldn't be alive. So membranes are a big deal. Non-membranous organelles are organelles that do not have a phospholipid bilayer associated with them. That would include the cellular cytoskeleton, which is made up of actin and beta-tubulin, microvilli, centrioles, cilia, flagella, and ribosomes. Ribosomes are made out of RNA, okay? Membranous organelles are all the organelles that have a membrane that looks actually a lot like the cell membrane. I wouldn't say identical, but really similar. So that includes the mitochondria, the actual nucleus or nuclear envelope, if you want to think about just the outside, the endoplasmic reticulum, both rough and smooth, the Golgi apparatus, lysosomes, and peroxisomes. So we're going to look at a few pictures first. In figure 3.1 in your book, you're looking at a picture of a cell that's undergoing mitosis or cellular division in a lung cell from a newt of all things, right? You guys know what a newt is? Look them up. They're totally cute. Basically, the reason why they use newts to look at their lung cells is because the lung cells in newts are actually very similar to human lung cells. Newts are easy to keep in terrariums for study. They're great research tools. What they did was they stained this cell with a number of colorful dyes. The blue is showing you the actual chromosomes, all right? So these are the X-shaped chromosomes that carry all of the genetic information for the organism. In other words, that's the chromosomes of that's the cellular DNA, I'm sorry. The green stains, what we call mitotic spindles, and the centrosomes are right here and here. The mitotic spindles are made out of tubulin molecules. The red is the cell membrane, okay, right over here. So this is probably a lipophilic dye that reflects red or emits red. It also picks up part of the cytoplasm. I would guess that this particular dye is staining the inside and the outside of the cell membrane. So this cell is in anaphase of mitosis. Now it looks like it's just going into anaphase. It looks like it's just leaving metaphase. We'll get to that later. So I talked about how the cell membrane, I talked about it briefly, is made out of a phospholipid bilayer. To understand what that means, we first have to look at what a phospholipid is. Whenever you hear the term lipid, that tells you that that's a fat, okay? Lipids come in a few different versions. This phospholipid has two long fatty acid hydrophobic tails. Hydrophobic means that this does not mix with water, okay? No water. I know that we often say hydrophobic means water-fearing, but these are molecules. They fear nothing. They have no feelings, therefore there's no fear. But they do not mix with water. Now, up here, hydrophilic means that this portion of the molecule is okay with water. Okay, philic means to be like attracted to, loving, okay? Again, molecules have no feelings. This means that this top end of the molecule is okay with being around water. So What that means is that you have polarity along the length of this molecule, okay? So you have the no water pole down here. Basically, water molecules will not hang out over here. And then you have the area where water is okay. All right, water is all over the place here. The reason why that's possible is that water and phosphate get along. Okay, this phosphate group. Down here, these are fatty acids. Now, what are fatty acids? Fatty acids are chains of carbon bound to carbon, bound to carbon, bound to carbon, carbon. So each one of these points is a carbon atom, okay? Now, what's interesting about this particular structure, the way I'm going to draw it, is carbons are able to bond to 4 other atoms. Uh-oh, sorry about that. So what this terminal carbon does is it binds to 3 hydrogens, rounding out its valence shell, and each one of these bonds to 2 carbons. And when you have the maximum number of hydrogens associated with all the carbons, we call that saturated, okay? Saturated is a fatty acid chain that has the maximum number of hydrogen associated with the carbons. Yay, Dr. Val, how terribly interesting. Hold on a minute, guys. We're getting to why this actually is super interesting. Over here, we have something slightly different happening. You see that there is a double bond here. Each one of these are single bonds, the zigzags, single bonds holding the carbon to the carbon. Each one of these carbons, in addition, has a hydrogen and a hydrogen and a hydrogen and a hydrogen. But when you get here, these 2 carbons have a double bond to its opposite carbon. So now you only have 1 hydrogen for each one of those. Each one of these then have 2 hydrogens again. Carbon, hydrogen. I'm trying to keep this from getting too complicated. But the magic of a phospholipid, one of the magical things, is that when you look at these unsaturated fatty acids, what that means is that you have fewer hydrogens and double bonded carbons. Oh, yay, Dr. Val. Let's go back to the uninterested boringness about this. Well, I could see that, you know, just looking at the structure is kind of boring. Great. One has a double bond, the other one doesn't. Okay, swell. That double bond changes how the phospholipid behaves. What that means is if you have unsaturated fatty acids, here's the key. This allows for membrane fluidity and liquidity. Again, bad writing, sorry. So what does that mean? What does that totally mean? Let's make a better star here. I found my eraser. What that means is that unsaturated lipids are liquid at room temperature, okay? These are liquid, relatively liquid at room temperature. Saturated are solid at room temperature, okay? So what's the big deal? All right, butter is a saturated fat. At room temperature, butter is solid. Olive oil is an unsaturated fat, right? It's liquid at room temperature. Which one is better for you? So butter or olive oil? Olive oil is slightly better because of this kinked unsaturated fatty acid. What that allows your body to do is more easily process the fat. So here's a little bit of physiology for you. But in addition to that, it is not as sticky. It doesn't set up a hard structure like a plaque in an artery. Okay, saturated fats build plaques inside of arteries if we consume too many of them. So saturated fats like animal fats, butter, lard, those all set up in our arteries because they all have these long, straight fatty acids. So in an unsaturated, you have straight. In saturated. In unsaturated, you're always going to have at least one of these little legs kinked, which means that the lipid is going to be liquid and it's easier for your body to handle that. Why is this important for a cell membrane? Cell membranes have two layers of fat. We call it the phospholipid bilayer. This region down here with the fatty acid chains is hydrophobic, so there's no water there. On the outside, on the extracellular side, the outside, there's water, right? We have water in our tissues. Inside of the cell, there's water. So what this does is, again, generate polarity. You have polarity, hydrophobic, hydrophilic, but it also provides polarity across the cell membrane, okay? No water, water. And this allows the cell to separate environments, outside to inside. The inside space of a cell is very different. Different things happen in there. The other thing that's important about the unsaturated nature of phospholipids is that that means that the cell membrane is fluid. It basically means that you're not a walking stick of butter, okay? It sounds gross, I know. But basically, each one of our cells has mobility. The cell membrane has mobility. That allows for other things to be inserted in the cell membrane. So if we zoom out a little bit, here are the elements that you're going to find embedded in a cell membrane. Here's the phospholipid bilayer with its polarity, no water in the middle. You have water on the outside, you have water on the inside. Now you have these little portals. This is called a channel protein. Only certain things can travel in or out of these channels. Let's say potassium, for example, okay? Glycolipids, all right? Glyco for sugar, lipid for fat. These are fats that have little sugars on them. These are important because glycolipids and glycoproteins, actually, this is a protein with sugars on it, they attach to the outer surface of the cell, allowing the cell to be identified by your immune system so that your immune system doesn't attack your own cells. When your immune system does attack your own cells, that's called autoimmune disease, and unfortunately, our body will tear apart itself if it doesn't recognize itself. We have peripheral proteins. You'll talk about those in physiology. Integral membrane proteins, those are important. They help anchor the cell. One of the stories I tell my class when I'm usually in class is that when I was a kid, I grew up in a very poor community, and I ended up with a biology book I don't know why I got this biology book, but I was in elementary school, and I was very bored during the summer, and we only had like 3 channels when I was a kid, so there wasn't a lot of TV. My mom worked, so I actually sat around reading books quite often, and I read this biology book, and one of the things I found out was our cells are anchored inside the body, that we're made up of cells basically, and they're anchored, but I didn't know how they were anchored, and they're actually anchored through these integral proteins and other cellular joinings, but I didn't read that far, and so one night I got really scared when it was time to take a bath, because, you know, again, I was a little younger. It's time to take a bath, and I'd been reading this biology book that were made up of cells, and I was worried that my skin would melt and all my cells would fall out. So I actually got away with not taking a bath for 2 weeks. Yeah, okay, there you go. Fun story from Dr. Val. That actually doesn't happen because of these integral proteins and cellular joinings that we'll talk about later. Once I learned about those, I slept much better at night when it came to that. All right, so what does the cytoplasmic membrane do? It is isolation, a physical isolation. The cell membrane is separating the external from the internal environment. It helps regulate the exchange of material in and out of the cell. So only certain types of molecules are allowed in and out, not everything. And sometimes conditions have to regulate what things go back and forth. So you'll hear a lot about that in physiology. The cell membrane allows cells to communicate, and to have sensitivity to the environment around them. And then, of course, the cell cytoplasmic membrane is structural support for tissues. So let's talk about things passing through the membrane. Again, you're going to talk about this a lot more in physiology, but we are going to touch on it here in anatomy because you need to remember that the permeability of a membrane is possible because of the channels that are embedded in the membrane, and different cells and tissue types are going to have different channels. So some membranes are freely permeable. Anything passes through. Okay, some are just leaky, I guess you would say. Others are what we call selectively permeable. And it's the selectively permeable nature of certain cells that kind of let them do the job that they need to do. So like neurons are selectively permeable. Some things can get through the membrane via what we call passive transport. In other words, if you have a membrane of a cell, which I'm going to draw like this, and you have a channel, and you have, let's say, 200 grams of, let's say, sodium, just sodium ions here, and 100 grams of sodium here. You have more here than you have here, therefore it's going to flow downhill. Passive transport means that ions are going to flow down their concentration gradient from high to low until you reach equilibrium, which would be about 150 grams of sodium on either side. Some substances, however, require energy to be moved back and forth. We call that active transport, and those are called pumps. So let's talk about a few examples of passive transport. Diffusion is just the movement of material from high concentration to low down the gradient. I just talked about that. It occurs through the lipid bilayer or through channels, across channels. This is very often seen with oxygen and CO2. Every cell allows oxygen and CO2 in and out of the membrane because they're small. They don't carry a charge. In other words, like sodium has a charge, a positive charge, okay? Oxygen and CO2 just diffuse across the membrane. Facilitated diffusion is where molecules are moving down their gradient, but instead of going right through the lipid bilayer, they're going through channels. It's facilitated. So this example here would be a case of facilitated diffusion because there's a channel carrying sodium across. And you will typically see this with anything that has a charge because a carrier protein is necessary to get the stuff through. And then osmosis is the movement of water down its concentration gradient. It's just like regular old diffusion. It just is specific to water. You're going to talk about that a lot in physiology. And the channels that water goes through, because water is a polar molecule, are called aquaporins. Let me get my eraser. Aquaporins. Aqua for water, porins for holes, water holes. So here's just a couple of little examples. Here is diffusion through the cell membrane, like oxygen, small nonpolar molecules, no charge, they just diffuse across over time. Facilitated diffusion, you have something that allows a charged molecule to come through. And then carrier proteins, definitely. This is something that's very specific, carrier proteins. This is where a protein, maybe each one of these green dots would be, or an amino acid, let's say, it would be like, I don't know, methionine, would be pumped in. Although I don't want to say pump because this is, again, this is passive, okay? No energy is used. When we talk about the sodium-potassium pump, this does use energy, ATP energy. And what that means in this particular case is, in this example, there are 3 sodium ions that lodge into the inside of this channel. And once ATP attaches, it ejects those 3 sodium ions, and then 2 potassium ions lock onto there, and as the ADP and the phosphate are released, the potassium goes in. So you have a net outward flow of sodium and a net inward flow of potassium. And notice, these are both monovalent cations. And then osmosis. This is a weird picture. This right here is time. This is time 1 and time 2, okay? So what you have in the case of osmosis is the red is water. The purple, it could be sodium ions, okay? Just think that the purple is sodium because sodium's easy. You have very little sodium and mostly water here. So you have 1, 2, 3, 4, 5, 6, 7 sodiums. The rest are water. Here you have probably 20 sodiums and less water. So here you have more water. Here you have less water. Let's make sure we write water. What's happening is water is moving down its concentration gradient. So it's kind of like if you ever took music, a crescendo. You have more water here than here. The water is going to flow across the membrane. The red, not the purple. Here you'll notice 1, 2, 3, 4, 5, 6, 7. 7 sodiums to start with, 7 sodiums to end with. Sodium didn't move, water moved across the membrane until you had an equal spacing of sodium and water here. Sodium and water here. So what you're looking at is the diffusion of water down its concentration gradient across a selective permeable membrane. All right. And the selectively or semi-permeable nature of this membrane is allowing water to go across. You'll talk about this in much greater detail in physiology. However, just so you know, what that means is that if you have more water inside of, let's say, a red blood cell and less outside, and all the water moves out, the red blood cells crenate, okay? They shrivel up because the outside environment was hypertonic. If you have equal levels of water outside and inside, the cells are happy and healthy. We call that isotonic. If there is more water outside and less inside, water gets pushed in. The inside of the cell is, the outside of the cell is hypotonic. It's pushing water in. The cells lyse or explode. Here you are, less water on the outside. So this has a dramatic effect on the hydration levels of tissues. So the membrane permeability, very important. Now for active transport, again, this requires energy. Active transport includes transmembrane proteins, ion pumps, and so forth that use ATP. Endo and exocytosis are also active, all right? So we're going to go over this just really quickly. There are 3 forms of endocytosis. In this first one, it's called phagocytosis. A very large particle is surrounded by, we call these pseudopods, pieces of cell membrane. It buds off and forms what we call a food vacuole, and that's used, whatever's in there becomes digested and used for energy. Penocytosis sometimes is called cellular drinking. So phagocytosis is like cellular eating. Penocytosis is like cellular drinking, where small items are being brought in. Same way, but these are all just little tiny particles inside a fluid. And then receptor-mediated endocytosis. This is interesting because a specific item has to lock onto a receptor, which can also be called, well, let's just call it a receptor for now. The receptor, once it gets in touch with its target, which is called a ligand, cause endocytosis, and however many ligands associated with receptors go into this coated vesicle, and the cell does what it needs to with it. And that's, again, physiology. And these are just definitions of each. And a couple other examples. Pancreatic cells are really good at secreting things. This would be exocytosis. And so look at the number of secretory vesicles inside of individual pancreatic cells, the acinar cells, okay? What kind of electron micrograph do you think this is? Is it looking at the outside or the inner structure? I'll tell you right now, it's the inner structure, okay? That means transmission. So let's look at a basic cartoon of what a human cell looks like. You will need to know each one of these elements for the exam, okay? And basically what they do, not the specifics. But we have microfilaments, which are a part of the cellular cytoskeleton, lysosomes, which secrete stuff, smooth endoplasmic reticulum, which makes steroids, fats, secretory vesicle letting stuff out. See, here would be the stuff, and the stuff is leaving, secreting. Peroxisomes, which inactivate things. Here's another lysosome. A vacuole. Not all cells have vacuoles, but some do. Cytoplasm. Cytoplasm, let's remember, cytoplasm equals the cytosol, which is the gooey liquid inside of the cell, plus all organelles. So if we're talking about just the liquid inside of the cell, that's the cytosol. But the cytoplasm includes the cytosol and all organelles. I'll let you guys go through the rest of the organelles yourself. What I'm doing here is showing you each element: endoplasmic reticulum, smooth and rough, Golgi apparatus, mitochondria, a peroxisome, the components of the cellular cytoskeleton, really beautiful structure, look at that. The nucleus and what lives in there. I'll stop here on the nucleus real quick. Chromatin. For the longest time, I would say, yeah, DNA is in the nucleus, but I'd always see chromatin, but I'm like, but DNA is supposed to be in there. What is it? Chromatin is DNA plus packaging proteins. What are the packaging proteins? Histones. Histones and scaffolding proteins, but we're not going to get into those details right now. That's a different class. But chromatin is DNA and all the proteins that package it up into chromosomes. So there you go. There's your DNA. Here's an example of a cell that has multiple nuclei in skeletal muscle. Here is an individual muscle cell right here. They're showing you a muscle cell right here. They offset the color. Each one of these spots is a nucleus, and you'll see on these muscle cells that an individual cell can have many, many nuclei. And the reason for that is that muscle cells are as long as the muscle, right? So think about some of the muscles in your legs, how long those muscles are. Each one of those cells requires protein synthesis to occur, which means RNA needs to be produced. If that happens, then how's the RNA going to get to all the way to the tip of that long, long cell if the nucleus is in the middle? So multiple nuclei accomplish the task of ensuring that protein synthesis occurs at the appropriate levels across the span of the cell. Cool, huh? Here's an example of a red blood cell in humans. So, and in all vertebrates, it turns out. Red blood cells are the one cell in the vertebrate body. I mean, there are a couple other examples, but they're not common. That does not have, a mature red blood cell does not have a nucleus. And it's because the nucleus is shed before the red blood cell enters circulation. And that increases the oxygen carrying capacity of a red blood cell. You lose the nucleus, the blood cell can be flat. It also changes what the red blood cell is able to do. It no longer makes its own new proteins. I mean, it does have some messenger RNA, but a red blood cell doesn't do like work. It just floats around. So this is one of the evolutionary modifications that makes vertebrate, I should say mammalian, it's not even vertebrate, mammalian red blood cells special. If you look at birds, guess what? They have nuclei in the red blood cells. Crazy, right? Dinosaur inheritance of birds. All right, so we're going to now move on to how cells are held together. Remember that crazy story I told you about thinking I was going to melt in the bath, right? Probably didn't help that I watched The Wizard of Oz for the first time right around then too. Anyways, cells form attachments to other cells or other materials like a basement membrane or adjacent tissue like muscle, using one of three major types of joinings or junctions. These junctions can be temporary or permanent. In many tissues, they're permanent, but we're going to explore tight junctions, gap junctions, and desmosomes. Okay, tight junctions occur near the tops of cells. Okay, so what we're looking at here is a picture of probably the digestive epithelium in the small intestine, where you have cells that the nucleus is down, the columnar cells, where the nucleus is down near the bottom of the cell, near the basement membrane. There are microvilli extending up on the surface of the cell. These microvilli are important for absorbing nutrients, okay, into the cell. And then these tight junctions occur at the base of the microvilli between cell 1, cell 2, and cell 3, okay? The adhesion belt is a region where you basically have shared tissue, shared actin, generating structure and strength in the membrane and in the tissue itself. And you'll see that these actin filaments extend up through each individual microvillus. We have button desmosomes about halfway down the cell. We have gap junctions halfway down the cell as well. These gap junctions allow items to go back and forth between cells, but we'll talk about that in a minute. So tight junctions are the ones up high. They have interlocking junctional proteins, which basically act like little spot welds between the two cells. You cannot separate these cells easily. And then you have the adhesion belt associated with the adhesion webbing here, okay, the terminal web. The adhesion belt is a place where you, again, have a nice tight junction or joining between the two cell membranes and a joining with the adhesion belt. So this blocks water, okay? No water can passes, okay? It also blocks other things from being transported from one side of the membrane to the other. Now, why is this important? Let's say you eat peanuts. What's underneath these cells? The bloodstream, okay? This is a blood vessel, okay? The way we absorb our nutrients is peanut protein, let's say, has to be transported into the cell. Things don't leak through. Things are transported through the cell and secreted out into the bloodstream. That's called transcellular transport. So when you eat peanuts, peanut protein goes through the cell, is processed by the cell, and amino acids are released into the bloodstream. AA is amino acid, okay? Peanut protein. So peanut proteins are broken down by digestive enzymes in our digestive tract, and elements of those proteins are absorbed by cells. Inside, the protein gets ripped up and amino acids are secreted. If, on the other hand, proteins leak through to the bloodstream between cells, that's when whole proteins get into the bloodstream and white blood cells start saying, hey, there's an invader in here. This is what generates food allergies. So it is very, very important that these tight junctions prevent the movement of items between cells into the bloodstream, okay? Because that will generate food allergies. So this can sometimes be an issue in young children. Sometimes when children are born, these tight junctions aren't fully formed. So as a baby is drinking formula or nursing, proteins get into the bloodstream and they start having allergic reactions. Now, gap junctions, on the other hand, are proteins called connexins. They look like little flowers on the outside that are basically tunnels that allow water and other chemicals across to the opposite cell. This also allows the movement of ions. They're big enough for, let's say, sodium to go across and potassium to go across. So it allows movement of small ions and small elements, not elements, I shouldn't say like chemical elements, but small items to go between two cells. Desmosomes, on the other hand, connect cells to other cells deeper down, and they also allow cells to anchor to the basal lamina. The basal lamina is kind of a layer of connective tissue underneath the cells, okay? Basically, you have a situation where you have these CAMs, cell adhesion molecules, that lock the cells together. And then the cell adhesion molecules are associated with this dense area here, this plate, this dense plate. And the dense plate, or dense area, we called it dense plate when I was in school, attaches to intermediate filaments, actin filaments holding the cells together, or attaches to laminins. Don't worry about what those are right now. They're proteins that hold these dense areas down onto the basal lamina or the basement membrane. All right, we're going to move on to the life cycle of the cell now. We're going to talk about mitosis, not so much meiosis right now. We'll talk about meiosis when we get closer to the reproductive system. Cells themselves can divide to produce, hopefully, identical copies of themselves. They clone themselves, and that process is called mitosis. And it happens after an egg is fertilized, so that mom and dad's genetic information contributes to making a zygote, an individual. And basically, from that single fertilized egg, an adult organism can develop. So we all grow through mitosis. We increase cell number through mitosis. We can also replace old or damaged cells and tissues through mitosis as well. So mitosis has basically a few different steps. Now, one of the things, I'll show you another slide of this in a minute, but one of the things, oops, my apologies, that I want to identify is, and this is the way I draw it, and the next slide is going to look a little different. We have 4 phases, G1, S, and G2 of the cell cycle, and M, okay? This is the cell cycle, okay? Not mitosis. M, that is the mitotic cycle. So mitosis, M-I-T-O, that was an O, not an E. Mitosis is a phase of the cell cycle. Okay, and mitosis is broken down into 4 phases and ends with a 5th phase. So mitosis is prophase, metaphase, anaphase, telophase, and ends with cytokinesis, cyto for cell, kinesis for dividing or pinching, where the cell divides into 2. G1 through G2 is all called interphase. And that's because early on in the 16 and 17 and even in the 1800s, you couldn't see if a cell was in G1, S, or G2 using a microscope, but you could see M. So M was well defined as prophase, metaphase, anaphase, telophase, and cytokinesis. G1, S, and G2 was just called interphase because they couldn't tell what was happening in the nucleus at those times. Here's a different picture showing you kind of how long each one of these stages takes. G1, the cell just kind of sits there until it gets the trigger to divide. And that would be right about here. This is called the G1 checkpoint. The cell basically hangs out and just does its job until it hits the G1 checkpoint. S phase, new DNA is made. New DNA is synthesized, hence S phase. G2 is a period of time where the cell is basically putting on a little weight, a little cytoplasm before it divides, maybe making up new organelles. The centrosomes are migrating to opposite poles. But very, very importantly, the new DNA that was synthesized during S phase is checked for spelling errors and mutation, and those errors are fixed. You would be surprised how often our errors are fixed, and actually there are very few errors that are made. So when those errors do get through, and there is a mutation, it's actually kind of rare. M phase is broken into the prophase, metaphase, anaphase, telophase, and eventually cytokinesis right here. So remember P, M, A, T, pro, meta, ana, telophase, PMAT. And what we're looking at here is just the definitions for each one of these phases and some pictures. Okay, prophase, metaphase, anaphase, telophase, cytokinesis. And at the end of the whole process, if the cell does not go through mitosis again, the cell undergoes differentiation. And so what we're looking at in this figure is the differentiation or hematopoiesis of individual types of blood cells. So we have a plasma cell here, a T lymphocyte, a B lymphocyte. This is figure 3.34 in your book. So you can look at this as an example of the ultimate outcome of mitosis and differentiation in a living set of tissues. Okay, I'm relying on you to go on ahead and review those figures that I kind of went through really quickly. You do need to know all of the intracellular organelles for the lab exam. You also need to know the stages of mitosis and what the chromosomes look like, how they're organized during mitosis. You should also be able to discriminate between a cell that is in interphase versus in pro, meta, ana, telophase, or cytokinesis.